The genetic history of Europe is complicated because European populations have a complicated demographic history, including many successive periods of population growth. The history must be inferred from the patterns of genetic diversity across continents and time. The primary data come from sequences of mitochondrial, Y chromosome, and autosomal DNA from modern populations and, if available, from ancient DNA. History and limitation of genetic studies. Classical genetic markers. One of the first scholars to perform genetic studies was Luigi Luca Cavalli Sforza. He used classical genetic markers to analyze DNA by proxy. This method studies differences in the frequencies of particular allelic traits, namely polymorphisms from proteins found within human blood. Subsequently, his team calculated genetic distance between populations, based on the principle that two populations that share similar frequencies of a trait are more closely related than populations that have more divergent frequencies of the trait. From this, he constructed phylogenetic trees that showed genetic distances diagrammatically. His team also performed principal component analyses, which is good at analyzing multivariate data with minimal loss of information. The information that is lost can be partly restored by generating a second principal component, and so on. In turn, the information from each individual principal component can be presented graphically in synthetic maps. These maps show peaks and troughs, which represent populations whose gene frequencies take extreme values compared to others in the studied area. Peaks and troughs, usually connected by smooth gradients, are called clines. Genetic clines can be generated by adaptation to environment, continuous gene flow between two initially different populations, or a demographic expansion into a scarcely populated environment, with little initial admixture with existing populations. Cavalli Sforza connected these gradients with postulated prehistoric population movements, based on archaeological and linguistic theories. However, given that the time depths of such patterns are not known, associating them with particular demographic events is usually speculative. Direct DNA analysis. Studies using direct DNA analysis are now abundant and may use mitochondrial DNA, the non-recombining portion of the Y chromosome, or even autosomal DNA. mtDNA and rydNA share some similar features, which have made them particularly useful in genetic anthropology. These properties include the direct, unaltered inheritance of mtDNA and rydNA from mother to offspring and father to son, respectively, without the scrambling effects of genetic recombination. We also presume that these genetic loci are not affected by natural selection, and that the major process responsible for changes in base pairs has been mutation. The smaller effective population size of the NRY and mtDNA enhances the consequences of drift and founder effect relative to the autosomes, making NRY and mtDNA variation a potentially sensitive index of population composition. These biologically plausible assumptions are not concrete. Rosser suggests that climatic conditions may affect the fertility of certain lineages. The underlying mutation rate used by the geneticists is more questionable. They often use different mutation rates, and studies frequently arrive at vastly different conclusions. NRY and mtDNA may be so susceptible to drift that some ancient patterns may have become obscured. Another assumption is that population genealogies are approximated by allele genealogies. Barbujani points out that this only holds if population groups develop from a genetically monomorphic set of founders. Barbujani argues that there is no reason to believe that Europe was colonized by monomorphic populations. This would result in an overestimation of haplogroup age, thus falsely extending the demographic history of Europe into the late Paleolithic rather than the Neolithic era. Greater certainty about chronology may be obtained from studies of ancient DNA, but so far these have been comparatively few, whereas Y-DNA and mtDNA haplogroups represent but a small component of a person's DNA pool. Autosomal DNA has the advantage of containing hundreds of thousands of examinable genetic loci, thus giving a more complete picture of genetic composition. Descent relationships can only be determined on a statistical basis, because autosomal DNA undergoes recombination. A single chromosome can record a history for each gene. Autosomal studies are much more reliable for showing the relationships between existing populations, but do not offer the possibilities for unraveling their histories in the same way as mtDNA and RY-DNA studies promise. Despite their many complications, genetic studies operate on numerous assumptions and suffer from methodological limitations, such as selection bias and confounding phenomena like genetic drift. Foundation and bottleneck effects cause large errors, particularly in haplogroup studies. No matter how accurate the methodology, conclusions derived from such studies are compiled on the basis of how the author envisages their data fits with established archaeological or linguistic theories. Genetic studies of Cavalli-Sforza. Relation between Europeans and other populations. According to Cavalli-Sforza's work, all non-African populations are more closely related to each other than to Africans, supporting the hypothesis that all non-Africans descend from a single old African population. The genetic distance from Africa to Europe was found to be shorter than the genetic distance from Africa to East Asia, and much shorter than that from Africa to Australia. He explains, both Africans and Asians contributed to the settlement of Europe, which began about 40,000 years ago. It seems very reasonable to assume that both continents nearest to Europe contributed to its settlement, even if perhaps at different times and maybe repeatedly. It is reassuring that the analysis of other markers also consistently gives the same results in this case. Moreover, a specific evolutionary model tested, i.e., that Europe is formed by contributions from Asia and Africa, fits the distance matrix perfectly. In this simplified model, the migrations postulated to have populated Europe are estimated to have occurred at an early date. But it is impossible to distinguish, on the basis of these data, this model from that of several migrations at different times. The overall contributions from Asia and Africa were estimated to be around 2/3 and 1/3, respectively. This particular model used an out-of-Africa migration 100,000 years ago, which separated Africans from non-Africans, followed by a single admixture event 30,000 years ago leading to the formulation of the European population. The admixture event consisted of a source population that was 35% African and 65% East Asian. However, the study notes that a more realistic scenario would include several admixture events occurring over a sustained period. In particular, they cite the spread of farming from a source population in West Asia 5000–9000 years ago that may have played a role in the genetic relatedness of Africans and Europeans. Since West Asia is sandwiched in between Africa and Central Asia, the model assumed an out-of-Africa migration 100 kya and a single admixture event 30 kya. However, most contemporary studies have more recent dates that place the out-of-Africa migration 50 to 70 kya. The study also involved a direct comparison between sub-Saharan Africans and Europeans. North African populations were omitted from the study. These considerations might help explain the apparent short genetic distance between Europeans and Africans. European population substructure or geneticists have found that Europe is relatively genetically homogeneous, but distinct subpopulation patterns of various types of genetic markers have been found, particularly along a southeast-northwest line. For example, Cavalli-Sforza's principal component analyses revealed 5 major clinal patterns throughout Europe. and similar patterns have continued to be found in more recent studies. A cline of genes with highest frequencies in the Middle East, spreading to lowest levels northwest. Cavalli Sforza originally described this as faithfully reflecting the spread of agriculture in Neolithic times. This has been the general tendency in interpretation of all genes with this pattern. A cline of genes with highest frequencies among Finnish and Saami in the extreme northeast, and spreading to lowest frequencies in the southwest. A cline of genes with highest frequencies in the area of the lower Don and Volga rivers in southern Russia, and spreading to lowest frequencies in Spain, southern Italy, Greece and the areas inhabited by Saami speakers in the extreme north of Scandinavia. Cavalli Sforza associated this with the spread of Indo-European languages, which he links in turn to a secondary expansion after the spread of agriculture, associated with animal grazing. A cline of genes with highest frequencies in the Balkans and southern Italy, spreading to lowest levels in Britain and the Basque Country. Cavalli Sforza associates this with the Greek expansion, which reached its peak in historical times around 1500 BC but which certainly began earlier. A cline of genes with highest frequencies in the Basque Country, and lower levels beyond the area of Iberia and southern France. In perhaps the most well-known conclusion from Cavalli Sforza, this weakest of the 5 patterns was described as isolated remnants of the pre-Neolithic population of Europe, who at least partially withstood the expansion of the cultivators. It corresponds roughly to the geographical spread of rhesus-negative blood types. In particular, the conclusion that the Basques are a genetic isolate has become widely discussed, but also a controversial conclusion. He also created a phylogenetic tree to analyze the internal relationships among Europeans. He found 4 major outliers: Basques, Lapps, Finns and Icelanders, a result he attributed to their relative isolation. Greeks and Yugoslavs represented a second group of less extreme outliers. The remaining populations clustered into several groups: Celtic, Germanic, Southwestern Europeans, Scandinavians, and Eastern Europeans.